Good morning. We're going to talk a little bit about cell types today. So there's several different cell types that are found inside plants. We're going to talk about these 7 types: parenchyma cells, collenchyma cells, sclerenchyma cells, tracheids, vessel elements, sieve tube elements, and companion cells. Last 2 kind of go together, so alrighty, so the first one we're going to discuss is the parenchyma cells. These are generally polyhedral in shape. They can be rectangular, kind of more, I would call brick-like, versus columnar, which might be a little bit taller, or elongated, which would be really, really long. Parenchyma cells at maturity are going to have a living protoplast. So they're going to have a nucleus, organelles, all those things. They're still technically living. These also generally have a very thin primary cell wall. Some of them might have a secondary cell wall. They generally will also have some sort of plastid, so a chloroplast or some other plastid. Like a chromoplast or something else. So the main job of these particular cells is to act as storage. They can be used for secretion. In some plants that produce nectar, these particular cells will form the nectary, which is the gland that produces nectar. So they would be used for secretion, not necessarily for storage. I said up there that they generally have that cell wall, but it's not always going to be uniform in thickness. Some parts of it might be thicker than others. In your first diagram that I drew, you can kind of see how parts of it are thicker than other parts. Parenchyma cells are normally going to have a larger vacuole for water storage, holding other dissolved nutrients or minerals, sugars, wastes. Parenchyma cells are often called totipotent because they have the ability to differentiate into a mature plant cell, or sorry, just a mature plant. So you can take one parenchyma cell and it can eventually develop and differentiate into other cells, which would eventually become an entire plant. So you can start with one parenchyma cell and end up with a full plant. It's called totipotent. And like that implies, your parenchyma cells can remain parenchyma cells. They can stay that. They are usually found in the softer plant tissues, or they can differentiate into becoming collenchyma cells. They can become sclerenchyma cells also. Your collenchyma cells are also going to have a living protoplast, so they're still going to be living with the nucleus and organelles. They're also going to have a primary cell wall, but this one's not going to be uniform in thickness either. In your diagrams, I've drawn you 2 different examples because sometimes they can have additional cell wall built on the sides or on the corners, as those 2 pictures denote. Their main function for collenchyma cells is to give additional support and structure to your cell. Think of celery. Most of the celery is going to be made up of parenchyma cells. But you know when you bite into celery and you have to bite through those ribs, those long, really thin tubes that kind of are located on the outside end there, those are collenchyma cells or collenchyma tissue. So that's usually you can bite through all the parenchyma cells fairly easily, but then you have to kind of rip that little rib, those little ribs that are going all the way through, and that's collenchyma tissue. So with your collenchyma cells, your cell wall is formed in layers. You generally start the first layer with a heavy or a thick layer, and then you get another layer. The next layer is more heavy with the pectin, that glue, and then it's going to alternate back to the heavy layer and then back to the layer with pectin. And so what that's going to do is give you more support. That switching of the layers gives you additional support. You can also have sclerenchyma cells. Overall, these have a thin primary cell wall, but they're going to have a thicker secondary cell wall. I don't want to get too far ahead, but they've got two different types, sclerids and fibers, but in both of these cases, you get a protoplast, which is dead at maturity, so there is no nucleus. It's kind of like a red blood cell. They don't have a nucleus either when they're mature. So what happens is the protoplast that was there leaves behind a very, very small space, which is called a lumen. It's not very big. With these cells, with your sclerenchyma cells, you're going to have these cells reinforced with lignin. The lignin is there to act as a barrier. It says for invasion, just keeping things out, basically. Keeping things out that you don't want. Like I said, you've got two different types. You've got the sclerid and the fiber, which I've drawn two pictures of those on your doodles. So your two different types, are going to talk just briefly about those two different types of sclerenchyma cells. We've got sclerids. You can have different versions of sclerids. They come in different sizes and shapes. You can have brachiosclerid, you can have astrosclerids, osteosclerids. So you can have different types of sclerids. These very much have, it's the first doodle on the left there, the second row down. You've got you can kind of see that the primary cell wall kind of goes around and then kind of interweaves inside and breaks up that secondary cell wall. So, and then you can see that the secondary cell wall is thicker than the primary cell wall, and then that tiny little space there is where the protoplast used to be, so that's the lumen that's been left behind at maturity. So this is reinforced with the lignin, very much so, and you're often going to find these in nutshells or fruits and seeds. Think of the hard kind of outer coating that seeds and nuts produce on the outside. So that's kind of what you're to kind of give you a real-world application there where you're going to see it. The second different type of sclerenchyma cell or tissue is going to be a fiber. Just like with your sclera, they're going to have a thin primary cell wall and a thick secondary cell wall. But instead of having these primary cell wall interweaving kind of throughout, it's just located on the edges. I've drawn you, because they're long or needle shaped, and then I've drawn you that cross section of it. So you can see that it's got a very thick cell wall, secondary cell wall, very thin primary cell wall, and that tiny space there where the protoplast used to be is the lumen. So you can see that both the scleroid and the fiber do have very small lumen spaces where the protoplast used to be. And then the main function for a fiber is going to be support. Your tracheids is the fourth type of cell type that we're going to talk about. These next ones are going to be similar. Your tracheids, vessel elements, and sieve tube elements, they're all going to kind of be similar at first glance physically, but they're all slightly different physically. And then they're obviously going to have different jobs. Your tracheids and your vessel tube elements are going to be very similar, but your sieve tube element's going to be a little bit different. So with your tracheids, you're going to have a thin primary cell wall, a thick secondary cell wall. You're going to find this mostly in your xylem tissue. They're also going to have lignin when they're mature. We call that being lignified at maturity. So again, they're going to have some of that lignin reinforcement for barrier protection. They are also dead at maturity, so no nucleus, organelles, things like that. So the empty space that used to be taken up by the protoplast allows water to travel through. So that's why these are found in the xylem tissue, because xylem is responsible for transporting water. And you can kind of see in the doodle that I have there. And then up and down these tracheids, you're also going to have these little holes, I guess. Well, it's like what we talked about yesterday, I believe it was. I filmed these out of order, so I think it was yesterday, about the pits. These are going to have bordered pits. So they're going to be those the pits that are going to have the secondary cell wall kind of curving around the pit. So those are going to be located up and down the tracheid itself. They have to be overlapping. That's how the water is going to be able to transport from one tracheid to another. You can kind of see that. I've drawn several different tracheids, right, kind of forming a long line, and you can see that they have to be, the bordered pit of one tracheid has to be aligned with the bordered pit of another, so that way the water can continuously flow through the xylem. Your vessel elements are similar to your tracheids. They do just physically how they look, and then also with their function. So they're also lignified at maturity. They're dead at maturity, so you're going to have that big hollow inside where the protoplast used to be. The main difference between your tracheid and your vessel element is there is no end. No end wall at the top and bottom. So what I was told to think of it as the toilet paper tube. that's left behind when you're done with the toilet paper. That's basically what your vessel element looks like. And so in order, instead of having to be laid on top of one another like your tracheids did, these just literally get stacked on top of each other to make one long tube or pipeline to transport the water. We'll talk about this more. I don't know if we'll talk about it tomorrow after your quiz or if it'll be on Monday. But just so you know, vessel elements have what are called perforated end walls. We'll talk about the different types of end walls that you can have. But just so you know, they have perforated end walls, in case you're wondering. So if you are looking at your tracheid and your vessel element right next to each other, the easiest way right off the bat is to look at their ends. If it's got kind of an open end or that perforated end, it's the vessel element. If it's got a full-on closed-off end, there is definite end tissue, then it's going to be a tracheid. So your sieve tube elements are, I'm not going to call it your last one, but they're almost like your last one because this and the companion cell work together. So first we'll talk about your sieve tube elements, and then we'll talk about the companion cells that are formed alongside your sieve tube elements. So with your sieve tube element, sometimes you'll see it called a sieve tube member. These are somewhat elongated, so they're kind of long and thin. They have a primary cell wall, no secondary, and it's primarily made out of cellulose. Unlike your tracheids and your vessel elements that can, and your sclerenchyma cells, you're not going to have lignin in this particular cell type. Sometimes they're going to have a couple of plastids, like your chromoplast, chloroplast. You're going to have some mitochondria. You're going to have lots of smooth endoplasmic reticulum, but you're not going to have too much else. I probably should have switched the order here. While they do have those particular elements, they do not have a nucleus when they're mature, which will be important, which is why you have a companion cell for these guys. Much like your tracheids, they have to be stacked on top of each other at the sieve plates. So if you look at the picture that I've drawn there of the sieve tube element, you can see the sieve tubes, or sorry, the sieve plates. And those little small dots in the plates are the pores, and that's where the food, generally it's the sugar, is able to move through these elements. So that's how they're going to be able to pass the food from one sieve tube element to the next. So these are generally going to be found in your phloem since they're responsible for transporting food and sugar. Okay. Your companion cells are formed alongside your sieve tube elements. The last set of doodles will kind of help you with this one. Initially, your parenchyma cells are going to undergo mitosis and cytokinesis. So they'll go through nuclear division, and then they will split the cytoplasm. You'll get the plasmodesmata forming, the pectin layer there in between. And one side is going to become a sieve tube element. And then the other side becomes the companion cell. The reason why is since your sieve tube element doesn't really have a nucleus or lots of functioning organelles, it needs that companion cell. Your companion cells are going to have a primary cell wall, and all of the necessary organelles that the sieve tube element and companion cell would need to function. The companion cell can then secrete substances either across the way into the sieve tube element, or it can receive substances that the sieve tube element passes on to it. So it's basically kind of like a mission control almost. So since it's got all of the organelles that can do all the processing and things like that, so that's kind of why these guys have their basic buddy, I guess. With your companion cells, they are very closely linked to that sieve tube element. When the sieve tube element dies, the companion cell normally dies as well. So they're very much linked together. So, like I said in the instructions, this will not be on your your quiz tomorrow, but we'll definitely include that on the next one. So if you do have any questions on these cell types or tissues, just let me know. All of these are individual cells, but obviously they can also be referred to as tissue. So you can have parenchyma tissue, tracheid tissue when you're talking about multiple of these cells working together. So just let me know if you've got any questions, and have a good day.